Hi, this is Mr. Warts again, and today we are going to focus our attention on a new module, DNA, which is found in the organelle, the nucleus. If you have any questions, by all means, watch the video again, refer to your book, or ask me in class. So the nucleus is the organelle that we're focusing our attention on. We've already talked about the cell membrane. We've already talked about the plastid. We've already talked about the mitochondria, and we are going to focus our attention now on the nucleus. And pretty much for the rest of the quarter, the nucleus is going to be it. Outside the nucleus, we find the nuclear membrane. It's double membrane, and it is continuous with the endoplasmic reticulum. Now, the nuclear membrane is very holy. It's also called the nuclear envelope. 25% of the membrane are actually pores. It is very selective what enters and what exits the nucleus. The nucleoplasm is the cytoplasm of the nucleus. Now, inside that we find chromosomes in the form of chromatin. The nucleolus is also found in the nucleus, but it's a high concentration of chromosomal loops. All those chromosomal loops get together, make a darker section, and that's what we call the nucleolus. The DNA is found in the form of chromatin, like I said before, in the chromosomes. So here's a diagram of the nucleus. We have the nucleolus here, we have the nucleoplasm there, we have the outer membrane, the inner membrane. Ribosomes are on the outside, not on the inside, and we have pores. Not enough pores, though. So there are 46 chromosomes in the Hugh people cell. 46. If we were to line them end to end, that is 12 centimeters of chromosomes. Now that really can't fit into the nucleus, not at all. They must be packaged. So we need to wrap the DNA around histone proteins. Now the DNA plus the histones are called chromatin. And if we were to take a look at it, you have 200 DNA bases wrapped around each histone. Actually a collection of proteins. We'll talk about that much later. And there's 50 DNA bases between the between the histones. I can even show you in class tomorrow. Now, each strand of DNA is a chromosome. So you actually have 46 strands of DNA in each of the cells. And here is, we have the nucleus, which was here, chromosomes here, which we unwrap. And chromatid is a copy of the chromosome. Beautiful. We see right here is the chromatin, the DNA, which is wrapped around the histone proteins. And we have the DNA, really kind of small. We can actually see the histones right here. That's a collection of 8 different proteins combined together. Again, we'll talk about that later on in the year. Now, DNA stands for deoxyribonucleic acid. The shape was discovered in 1953 by James Watson and Francis Crick. Maurice Wilkins lent a hand and also won the Nobel Prize with those 2 other gentlemen. Rosalind Franklin actually took this picture, which is called Photo 51 of DNA, X-ray crystallography. It is almost a perfect picture, which allowed Watson to realize that it was in the shape of a helix. It was a helical structure. And without this picture, Watson and Crick would not have discovered the structure of DNA. Unfortunately, she did not win the Nobel Prize because she won earlier of cancer. So she died. And they didn't give the Nobel Prize posthumously like they did this past time. So the basic unit is a deoxynucleotide. We have to say deoxy because nucleotides would be RNA. The phosphate right here, we know what phosphates look like, beautiful. We have a deoxyribose right here, and a deoxyribose is a deoxyribose because it's missing an oxygen right there. The nitrogen bases are either pyrimidines, which is cytosine and thymine, or purines, which is adenine and guanine. And here's a picture of the nitrogen bases here. The purines are double ringed. We're not going to talk about uracil, that's only in RNA, because we're going to focus our attention on thymine, which is found in DNA. Now, the only difference between thymine and uracil is this CH3 group here. The CH3 group here. There is no CH3 group there. That is the only difference. Now, if we put together the phosphates, the deoxyribose, as well as the nitrogen base, we have a deoxynucleotide. there are 3 phosphates associated with a deoxynucleotide. We break off 2 of those phosphates when we form a DNA chain, and that provides the energy necessary to make DNA longer. We have deoxy adenosine triphosphate, DATP. We have deoxyguanosine triphosphate, which is DGTP, deoxythymidine triphosphate, which is DTTP, and deoxy, this should be cytidine triphosphate, which is DCTP. Now, notice that they're all pretty much the same. We have the 3 phosphates, the deoxyribose, and the nitrogen base. So if we were to draw this, put it together, hopefully the next slide, nope, I'm sorry, we'll put it together in a moment. The deoxynucleotides, they will form covalent bonds between the deoxyribose, which is the sugar, and the phosphate group via synthesis reactions. So 2 phosphates are cut off of the deoxynucleotide, and that provides the energy so that the phosphate can add to the 3' end of the deoxynucleotide. What does the 3' end mean? And you're going to realize that there are actually 2 ends. There's going to be a 5' end and a 3' end, and that deals with the deoxyribose molecule, the number of carbon. This first carbon is the first 1'. This is the second. This is the 2'. This right here is the third carbon, which is a 3', 4', and the 5' end. We can only add DNA bases or deoxynucleotides to the 3' carbon down here. We can't add it to the 5' carbon. When this right here has no DNA bases above it, this is the 5' end. When there are no bases below this carbon, that is the 3' end. But you need to remember, and this is so very important, that DNA can only be added to the 3' carbon. It can only be added here, 3' carbon. So now you know about the 5' and the 3' end. So over here, we have the deoxyribose. We have the phosphate. This is the 5' end because there's no other deoxynucleotide above here. We have, and you maybe want to draw this, we have the sugar, we have the phosphate, we have the sugar, we have the phosphate, the sugar, the phosphate, the sugar, the phosphate, the sugar. Notice down here, there's no DNA bases below the 3' carbon here. So this is the 3' end, 5' end, 3' end, and the nitrogen bases are over there. Now this right here, which is a sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. This is known as the sugar phosphate backbone. That's what you form when you add the phosphate to the 3' carbon. So the phosphate would be added here, and we would have another base down here. DNA is double-stranded. We're going to put another strand to that sugar phosphate backbone. DNA is also anti-parallel, which means that that second strand is upside down. So you still have the sides of the DNA being the sugar phosphate backbone, but the nitrogen bases in the middle are bonded together through hydrogen bonds. And it is a very precise arrangement with adenine and thymine and cytosine binding to guanine. So adenine forms 2 hydrogen bonds with thymine, and guanine forms 3 hydrogen bonds with cytosine. This is known as Chargaff's rule, who discovered that the adenine and the thymine had the same ratios as the cytosine and guanine. Now, together, all this stuff forms a double helix, a spiral staircase, and not a twisted ladder. So let's take a look at the double-strand DNA. We have the 5' end here. We have the 3' end here. On the other side, we have the 5' end here and the 3' end up here. So the DNA is anti-parallel. This side is upside down when compared to this side. Off of the first prime carbon, we have adenine, first prime carbon, thymine, and they form 2 hydrogen bonds. We have guanine forming 3 hydrogen bonds with cytosine, thymine forming 2 hydrogen bonds with adenine, and cytosine forming 3 hydrogen bonds with guanine. Okay, again, we have the 5' end because there's nothing above here. There's no DNA bases above the 5th carbon. This is the 3' end because no DNA bases are attached to that. But if we were to add DNA, we would have to add it to the 3' carbon here and a 3' carbon up there. DNA is a double helix, not a twisted ladder. This is a twisted ladder. The middle, or the axis, goes right down the middle here, whereas this will wrap around the axis. So a spiral staircase moves around the center pole, where the center pole of a twisted ladder goes right down the middle of the ladder. It is not a twisted ladder. This is wrong. So you might be asking yourself, well, what holds DNA together? Hydrogen bonds. They are so important. We are here because of hydrogen bonds. Hydrophobic interactions. The DNA bases are ring molecules, so by definition, they are hydrophobic and base stacking. They have weak associations because the bases are stacked on top of each other. How do we denature DNA? In order to denature DNA, you need to break the hydrogen bonds. And we have learned how to break hydrogen bonds. We increase the temperature so the hydrogens vibrate. We decrease the pH so the hydrogen ions are attracted to the partially negative ions. We also can increase free radicals so we can have the negative OH groups attracted to the positive hydrogens. So those are the ways that we can denature DNA. The next screencast, we're going to talk about DNA synthesis and replication. But hopefully you understand all the basics behind DNA. Hope that helps.